All right, so I'm happy to announce the students who will be presenting for MCDB 1161. They're Charlotte Georgemans and Kate Schinkel, and they'll be talking about discovery of and isolation of bacteriophages.
Awesome. Thank you, Dr. Philman. I will present my screen. I am not able to present my screen. I don't know if I am able to present slides.
Oh, it's this, it's share screen. We have to give permission for multiple users. Pam, is that you? Do you do that?
Yeah.
Okay.
Charlotte, I'm going to make you a host, okay, so that you can do this.
Sounds good. Thank you.
You should be good at this point.
Awesome. Thank you. All righty. Can everyone see my screen? Awesome.
Looks good.
Well, hi, everyone. It's good to meet you all. My name is Charlotte, and today I will be presenting with Kate Schinkel, and we will be presenting about our experience in MCDB 1161. MCDB 1161 is a semester-long course offered at CU where we collect soil samples and isolate bacteriophage. So a bacteriophage is called a phage, and phage are viruses that infect and replicate within bacterial cells. And so phage can be classified in two different categories. There are lytic phage and temperate phage. So lytic phage follow the lytic life cycle, where a phage attaches to a bacterial cell and inserts its DNA, and within the host cell, the new parts of the phage are created and they assemble together, and the host cell lyses, releasing these newly synthesized phage. Now, temperate phage follow the lytic and lysogenic life cycles, where in the lysogenic life cycle, the DNA from the phage is directly integrated with the bacterial cell's genome. And the lysogenic cycle is exited via certain environmental conditions, and the phage will then go into the lytic life cycle. So phage structure is very important. Here's a diagram of what phage look like. So in the head right here, it's called the capsid, and this is where DNA is held. And the tail is a very integral part of the phage, as it helps to transport the DNA from the head into the bacterial cell. And we also have tail fibers and spikes, which are very important because they help the bacteriophage kind of recognize host cells that it can bind to and replicate. And there are also three morphotypes of bacteriophage. So meet Gortil. Gortil is a Sifoverde phage. This is actually the phage that my lab partner and I isolated together. And as you can tell, Gortil has a very long tail. It's known to be non-contractile, and the head looks pretty normal, like a capsid. This is Ricky, a Myoverde phage. The tail is shorter and non-contractile, and the head seems to be larger. Sifoverde and Myoverde are the most commonly isolated morphotypes of phage in our class and in our region. And finally, we have SH1, which is a Podoverde. They are known to be the smallest morphotype. So morphotypes are very important, as it contributes to biodiversity within phage, which can help with significantly improving and diversifying research and medical treatments. Now, I'll talk about plaque morphology. So a plaque is showing the presence of phage. So as we can see on this picture, we have this bacterial lawn, which is the opaque part. So that is the host bacteria that we put on our plate, which is called M-schmeg. And these clear areas are plaques, which basically show the presence of phage. Now, we can recognize that we have a temperate phage here because the inside is clear and the outside is more of a halo kind of shape. And also in this picture, we can see we have lytic phage because they are smaller plaques, but they are also completely clear throughout. And so knowing the type of phage that we have is very important, because that allows us to determine whether or not our phage is a good match for certain medical treatments, like phage therapy, which Kate will get into a little later in this presentation, as well as various applications of our phage research.
All right, thank you, Charlotte. So another way that we can categorize phage is by cluster. Cluster is going to group phages by similarities in their DNA genome. And one way that we can begin to predict this is with restriction digest. So as you can see here, we have restriction digest where the phage genome was cut by 6 different enzymes. And this will produce a specific cut pattern. This is often similar between phages of the same cluster. And this is where we can really kind of begin to look at other restriction digests with similar cut patterns and begin to predict the cluster. In order to support this prediction, we can use PCR and specific cluster primers, which are able to support this a bit more. As you can see in this photo over here of the PCR, we have a B1 primer, and this does indicate presence. It's important to note, however, that until we sequence the phage genome, we can't actually know the cluster, but this gives us a pretty good guess. Cool, thank you. And then, so why study phage anyway? Well, antibiotic resistance is becoming a more and more pressing issue in our world. As we all know, bacteria randomly mutate, and in the course of antibiotic treatment, some of these mutations may prove useful in avoiding the antibiotics because these are the phage or the bacteria that survive. They're going to make up more and more of the population. What this means for us is that a lot of antibiotics that used to work no longer do. In fact, it's estimated that by 2050, infection deaths caused by these antibiotic resistant bacterial infections will reach 10 million annually. And we're only really creating an environment where this is more and more common. In 2020 alone, more than 10.5 million kilograms of antibiotics were used for food producing animals. But there is promise in phage therapy. One such case is Dr. Tom Pattison. So upon vacation with his wife and professor of epidemiology, Dr. Stephanie Strathy, Tom ended up catching an antibiotic resistant bacterial infection. And despite many different types of treatments, things were looking pretty dire, as you can see in that photo above. And Dr. Strathy advocated for use of phage therapy after the antibiotics were not working. Today, Tom is alive and healthy, and we've seen a lot more progress in cases like this for treatment with phage. UC San Diego School of Medicine has an institute that specifically dedicates research to phage therapy, and Dr. Strathy even came to give his talk here at CU. So the future of phage therapy is something that's constantly evolving, quite excitingly. One avenue is human medicine. So as we can see from these panels above, we have first a wound that wound treatment with antibiotics is not healing. A phage-infused bioderm is then applied to this wound, and we can see recovery by the end. So this is a really promising avenue for antibiotic-resistant infections that previously seemed not to really have too much hope. Another avenue is agriculture. This is where a lot of our antibiotics are used, and phage offer more specificity as well as the ability to conserve a lot of our antibiotics. Finally, there's research into reducing antibiotic-resistant bacteria by engineering phage that target the traits of these antibiotic-resistant bacteria, which is a really interesting way to look at how kind of this problem has evolved.
Awesome. Thank you, Kate. So one thing that I found very interesting in this class is how to apply what we've learned into different fields. So my background is not in MCDB. I'm actually in the College of Engineering, and I'm a computer science student. And so I learned about kind of a study happening at Brandeis University where researchers are currently making computational models that model kind of phage capsid assembly. So if we remember from the structure slide, the capsid of a phage is the head. So right here, and it's where the DNA is held within the phage. And so these researchers were able to use the research that they've conducted and all the data and compile that with programming to create this model. And they were able to kind of see how capsid protein surrounded the phage genome and predict kind of the steps involved and kind of simulate the assembly. And this is very important and very useful because it gives us another dimension and way of looking at how phage are assembled and made, but it also helps us understand kind of the misassembly probability and prevention, which is very important because if we are able to catch a like phage capsid being misassembled and prevent that, that can prevent us from accidentally including this phage in a phage cocktail or having possible experimental error in the future. And finally, we would like to thank the HHMI C-Phages program, our professors, Dr. Guild, Dr. Philman and Megan Greening, as well as our TAs, Nye and Brooke. I can speak on behalf of both of us that we were very grateful to have been in this class and be exposed to MCDB department. And here are our references. Thank you for your time.
If anyone has questions for our phage presenters, you can either throw them into the chat or feel free to raise your hand, or you can just unmute.
Hi, I actually had a question.
Go for it, Dylan.
All right, so you mentioned that phages are used to treat antibiotic resistant bacteria. Are there any cases where these bacteria can actually develop resistance to the antibiotics as well?
Do you mean to the phage?
Yeah.
Yeah, so this is often why antibiotics and phage are combined, because together it kind of creates this total wipeout of all bacteria. They do pose some of the same risks as we do in the overuse of antibiotics, but phage are often more specified as well, which can be helpful because it's not just like one broad antibiotic that tends to be overused or we have antibiotic resistant bacteria popping up. But yes.
Awesome, thank you.
And it looks like there's a question in the chat from Brandon. With learning the assembly of the phage capsid, could this be used to make synthetic capsids for future treatment?
I can answer this one. That's a great question. So that is something that the researchers are looking into. So this study was more on kind of the probability and the math kind of theory and computational theory behind capsid phage, like phage capsid development. But I can speak that that is definitely an option that they're considering. It's probably more far off in the future as this is fairly new research. But if we can create synthetic phage, that would be very beneficial, especially for phage therapy. We can currently modify lytic phage, or temperate phage. So if we are treating a patient via phage therapy, we would rather treat them with lytic phage rather than temperate phage, just based on the plaque morphology. So that is something that is currently happening, genetically modifying some phage.